The species comparison tool allows you to compare human pathways with other species to see what is common to both or perhaps missing in humans or in the model organism. Species comparison is launched using the Analyze Data button in the Pathway Browser header bar or the Analyze button at the top of the Pathway Browser. In the Species Comparison section, select one of the species in the drop-down list. Click the Go button. Reactome uses the manually curated human pathways to computationally infer their equivalence in other species. This inference process is based on gene orthology, using the Ensemble Compara database. Biological processes are remarkably well conserved over large evolutionary distances. Even though our focus is to annotate the molecular details of human biological events, we can use protein sequence orthology relationships to ask whether the human proteins involved in the reaction have orthologs in the model organism. If the orthologs exist in the Ensemble Compara resource, we computationally infer the corresponding reaction for the model organism and in this way build up a predicted pathway knowledge base for the organism. Clicking on pathway nodes displays the relevant pathway diagram. The color of reaction objects indicates the result of the comparison. Yellow indicates that the protein has an inferred equivalent in the comparison species. Blue indicates that no equivalent was identified. This protein may not exist in the comparison species. No color indicates that inference was not possible. This is always the case for small molecules, DNA and other objects that have no UniProt entry. Objects with bands of color represent complexes or sets containing more than one molecule. The bands of color reflect the inference success for the molecules within the complex or set. The species comparison bar at the bottom of the pathway diagram can be used to turn off species comparison coloring. To see the species comparison results for the components of a complex, hover your mouse over the object and open the information panel. Traditionally, reactions in Reactome represent events that occur within a single generic human cell. It would, however, be useful to classify reactions into different human tissue types, as to provide an evolving picture of the reactions and pathways in different cell and tissue-specific environments. This tool allows users to understand the pathways that are enriched in different human tissue types, based on protein expression data. Tissue distribution is launched using the Analysis button in the Pathway Browser header bar. In the Tissue Distribution section, select the one experimental tissue dataset, HPA, EPROT3, Expression Atlas, in the drop-down list. We have imported protein expression in different cell or tissue types from the Human Protein Atlas or HPA, overlaid these proteins on Reactome data, and extracted the subset of reactions for that particular cell type. The HPA data reflecting the expression of the protein coding genes in 44 different human tissues can be visualized through the analysis tools. Once the window refreshes, select the available tissues in the left panel, and hit the Add button to add tissue expression data to the analysis tool. Press the Add All button if you would like to adjoin all the tissue expression data. Use the Remove All and Remove buttons to delete tissue expression data from the filter list. Once you have selected all the appropriate tissues, click the Go button to start the analysis. An overrepresentation analysis is performed on the protein expression data of each tissue type. The results of the tissue distribution analysis are presented similar to that of overrepresentation. Users can use the slider at the bottom of the diagram panel to toggle between the selected tissue types. The Analysis tab in the Details panel contains the enrichment statistics, and the diagram panel displays reactions or pathways with overlaid tissue-specific expression data. There are several options available to export analysis results in various formats. The Downloads button is located at the left of the Analysis tab in the Details panel. The following files can be downloaded. Complete or filtered analysis results in CSV format. Complete analysis results in JSON format. Identifier mappings of submitted data and selected resource used for analysis in CSV format. A detailed report of the analysis for top 25 most significant pathways in PDF format. A list of submitted identifiers that were not found in CSV format. Analysis results overlaid on pathways and reactions can also be downloaded using the Export button in the diagram panel. This feature takes a screenshot of the current diagram panel, which can be saved in PNG, JPEG, SVG, or PDF formats. Analysis results on the Voronoi pathways view can also be downloaded using the Export button in the page.